Good evening, ladies and gentlemen. Can everyone hear me? Can you hear me up the back? All nice and clear? We've nearly made a full house. Thank you very much, everyone, for coming along tonight. I understand that it's raining a little bit. It's very cold. Hayden, can we just dim the lights a little, please? They're very bright. Thank you. Distinguished guests, ladies and gentlemen, welcome to Brain Research, Progress and Prospects. I'm Sue Giddens and I'm thrilled to open this very special evening. A little bit of housekeeping. Should we need to evacuate the building, please make your way to the nearest exit and follow one of the wardens who will have either a Brain Research t-shirt on or a security uniform. This event is part of the Neurological Foundation's Annual Appeal public lecture series. As you've seen, the TV ad from our TV campaign, which played through over the last week, featuring tonight's guest speaker. Our appeal campaign is designed especially to raise awareness of the critical need for brain research funding. I'd like to make mention of the very special 22-year relationship that we've had with Mitre 10. They sponsor the Foundation's annual appeal. They help to pay for our annual appeal envelopes, which I hope you'll all have taken as you made your way in. They pay for the print of the envelope, quite a lot of our marketing communications material, and help to spread the message across all of New Zealand. Quite simply, without Mitre 10 and their passionate staff, the Foundation would not be able to have Appeal Week. So thank you, Mitre 10. The Neurological Foundation is the only non-government neurological research funding body in New Zealand. Over 98% of funding comes from people like you, from members of the public and from community groups and fundraisers and donations that come in envelopes. The other 2% or so come from bodies such as private foundations and guardian trusts and so on. Funding for brain research is needed more than ever to improve the outcomes for the growing number of New Zealanders being diagnosed with neurological disorders. As our population ages, so too does the number of these disorders, and the only way to end the devastating neurological disease afflicting so many people is with the best research. Research is, however, making remarkable progress, as you'll find out tonight from our speaker. He has contributed hugely to the advancement of neurological research, and his passion and commitment to improving the odds for patients is significant. Home when Richard Faull and his four brothers were in short pants was a place called Tekorangi, a North Taranaki town comprised of a general store, a church and a school. Richard's parents ran the store. Neither completed secondary school, but passed on to their children an ethos founded on education, enthusiasm, determination and service. Richard says, "My mother was the eternal optimist. Ingrained in all of us was that you served your community. I had pretty humble beginnings insofar as Mum and Dad didn't have money, so we dealt with the principles of people. Mum always said, put yourself in the shoes of the other person, and always remember that if you complain about having those shoes, there are people who actually have no feet." Richard's path was sealed with the first glimpse of the human brain as a third-year med student. He says it was absolute love at first sight. During his 40-year career, Richard has made major contributions to scientists' understanding of the changes that occur in the brains of people with diseases such as Huntington's, Alzheimer's and Parkinson's diseases, and disorders such as epilepsy and schizophrenia, opening the way for the development of new treatments and one-day cures. He is perhaps best known for his research demonstrating that the adult brain contains stem cells. This groundbreaking work not so long ago countered the long-held belief that adults have a finite supply of brain cells that cannot be replaced once they have died, and offers hope to people with neurodegenerative diseases and to those with brain injury. The Centre for Brain Research, which was launched in 2009, is the realisation of Richard's vision for better research through partnerships between neuroscientists at the University of Auckland, New Zealanders affected by brain disease, and the neurologists and neurosurgeons who work with them. The centre houses the Neurological Foundation Human Brain Bank, which Richard founded in 1994, and the Brain Bank now houses, I believe, tissue from over 400 brains. The Brain Bank is the largest of its kind in the Southern Hemisphere, and it holds brain tissue donated by people with various neurological disorders. It is a crucial resource for neuroscientists in New Zealand, and it has generated numerous collaborations with leading brain research laboratories around the world. It is also an example of the strong links that Richard has forged within the community. Richard has been given numerous awards, including the Fellow of the Royal Society, the Officer of the New Zealand Order of Merit, the Lilly Medal, and New Zealand's top science honour, the Rutherford Medal. In 2010, he was named the Supreme Winner of the World Class New Zealand Awards, and in 2011, Richard was named Distinguished Professor of the University of Auckland. He has flown down from Auckland, especially for this event tonight. Please join me in welcoming a very special guest to the stage, Distinguished Professor Richard Faull.
Coming through? Thank you, Sue. My God, those lights are light. So it's an incredible pleasure to be here. I mean, Christchurch is a special place. It's a place of resilient people. And that's critical. You know, I spent a fantastic afternoon today talking to some of your top neurologists, some of your great scientists. And I know many of the brain scientists here, most of them personally in different ways. And you know what we're plotting? We're plotting a journey where we're going to combine together with the Otago neuroscientists, together with the Auckland ones, to attack brain research head on. Go for a big government grant after one of these centres of research excellence. So I've had an incredible afternoon, and it's been great. You know, when you meet people across the table and they've got the vision, the passion to push back the boundaries of research, that's marvellous. And so having the opportunity to talk to you tonight is actually a privilege for me. As Sue said, you know, how my mum and dad said, be humble, keep your feet on the ground and just get on and do the job. And that's what it's about. And tonight, you see, is a special night to come and tell you, take you on an exciting journey. A journey in brain research. Tell you some of the things we've done. I was joking to Sue before and I said, you know, it's actually a free lecture, right? I said, what we haven't told everyone is free to get in. We're going to lock the doors. It was $50 each to get out. No. Anyway, no, it's not like that at all. Because we want to give more. All right. So, so I've had an incredible life, and it's still going. It's getting more exciting. And one of the reasons is that I get paid to do something which I love, study the human brain. And one of the major reasons I've been able to do that is funding from the Neurological Foundation. For over 30 years, in one form or another, they have funded, as I'll show you as we go along, our research. And that has been marvellous. And so the fact that you people have come out tonight to show your support is something very special. So thank you for that. And so, you see, I want to take you on a journey of discovery tonight. Discovery on the human brain, and I've got to show you the human brain. I've got one bought especially for the occasion. So it's very special, you see, so I've got to put the cloth down. And my little bag of tricks here. But here I have the human brain, which has travelled almost as much as what I have. It's a brain which was bequeathed to us, like many have been over the years, by families to do brain research. And as Sue said, when I first saw this as a third-year medical student and saw all the folds on the outside, coming from a family background, I didn't know I was going to find at medical school, but I saw this brain. And we were given the incredible privilege in third year of spending every Saturday morning, believe it or not, for 10 weeks, and we dissected and looked at the human brain, found out what was inside, found out all the bits and pieces. And I wondered, how does this work? And so that's when I fell in love with it, and that's why I ended up doing brain research. And to do brain research, which is your passion and your vision in life, and get paid for it, has got to be incredible. To find out things you never expected. The journey I'm going to take you on, you see, is a journey which I never expected to travel. Which all my colleagues and people here tonight that we work with here in Canterbury, Christchurch, we didn't expect to travel. But it's incredibly exciting. We've got huge challenges ahead, but we've made lots of progress, but we've got so much more to do. And so you see, in the first slide you actually see, push the right button. Well, so in the second slide, there we are. That's the human brain. It's actually the brain that's sitting right here. It's incredible. And all these folds you see on the outside, the convolutions. Now, you know, this is what makes each one of you special. This is a gift which you get when you're born for free. And it represents who you are, what you are, your passions in life, your vision, your creativity. Every day you get up in the morning, you should say, first, thank you for my human brain. Because what you are and who you are, where you're going, where you've been, your vision ahead, depends on this organ. And that is incredible. And we understand a lot about it, but there's a lot we don't understand. We've got huge frontiers to push back. And in a way, it's almost an impossible task, because I always say that we really need a brain bigger than that to understand it, you see. But we're doing well. We actually are getting there. But as you'll see, it's a marvellous machine. And to actually, when you go to the next slide, you will see, and this is not quite going to, yes! You see, to appreciate the complexity of the human brain, I've lined up here a rat brain, a monkey brain, and a human brain. The rat brain is very special. It's what we call the prototype of the mammalian brain. It's got the smooth outer forebrain here. It's what I call the Model T. Because you see, as you go up through the animals, haven't got the cat brain here, but the cat brain is much bigger. And as you go up to the cat, to the monkey, to the human, it's this forebrain, you see, which gets bigger relatively faster and faster than the rest of the brain. And in order to accommodate this flattened cerebral cortex on the outside, begins to fold. And the more it folds, you see, the more brain cells you can fit in the front of your brain, your forebrain. So it's our forebrain here which makes us absolutely unique. You know, rats, cats and monkeys are doing the same today as what they did 1,000 years ago. You don't see them building libraries. You don't see them flying aeroplanes. You don't see them flying to the moon or whatever. Developing new IT machines. No. You see, humans have this unique ability that every generation advances in knowledge on the previous generation, and that's because of your forebrain. That gives us communication, verbal language. We write down, we read, we learn, we advance, you see. And that's because of the forebrain. Incredible. And so, you see those folds which you see on the outside of the brain, they're not random. There's actually a pattern to them. If I had time, I'd go through and I'd name them all for you. But there's actually a pattern, you see, coming down over the side. This is looking at the left side of the brain. There's a fold that comes down here, and that controls movement on the opposite side of the body. Movement in the most complex way. So if you sneaked in inside the brain, and I started stimulating here, around there, different muscles on your face and your lips would move. And if I stimulated up here, muscles which controlled your fingers, your arm, your forearm, your thigh here, your leg and so on. You see, the map is incredible. And if I cross the divide into the fold behind, you go into from the USA into Canada. Because you see, this is the area not of control of muscles, but sensation. What do I mean by that? Well, you can feel touch and pressure on any part of your body. You can feel someone gives you a passionate kiss, you feel on your lips. Well, actually, you don't. Where you feel it is here. Because this is the area of conscious sensation from your face. So you think you feel it on your lips, you're actually feeling it here. Because if there's a small tumour or a stroke which appeared there and destroyed this area, you'd be numb on your lips and face, depending on how much it involved. You see, the ability somehow that your conscious appreciation of sensation there is projected out to the area of the skin is something we actually learn. And so someone drops a brick on your foot, you feel it there at the top. You see a picture, this on the screen, you think you're seeing it on the screen, you are actually, but it's projected out there. You're actually seeing it here at the back and it's upside down. But we turn it over. We turn it the right way up because we've learned to do that. And it's there in the most intricate detail with colours added to it. Memory is stored here in the most intricate way. We've only got little clues about. The reading and writing, the ability is there, you see. The ability to speak is there. It drives the muscles in your throat which speak. And your personality and who you are and what you are, which is unique for every one of you. You're extrovert, outgoing, easygoing, whatever. You've got a personality, it's the same each day. Very much depends on this large forebrain. Our behaviour is complicated. We have ideas of imagination, of going forward. We have ideas of how to make our business better, how to farm better. Whatever it is, every one of you, you see, has a unique creative brain. It may look the same on the outside, but on the inside it works differently. You see, I can't look at this brain and say this is from a female or a male. I know it's from a mature person. I can't tell you whether that person had a bent towards farming, towards networking, IT skills or whatever. I can't tell anything about the personality. Einstein's brain didn't look any different to anyone else's brain from the outside. On the inside, they think there was a few more glial cells, but that's up for grabs anyway. So you see, it's incredible, isn't it? And that's your human brain. Now what's interesting though, is that all these different functions that I've talked about, they actually happen because there are brain cells underneath, just underneath the surface here. And I'll tell you a little bit about those. And there's at least 100 billion brain cells, all right? That's more than the stars in the Milky Way. And if I was to take a section just through the brain here, and go to the next slide, ignore this bit here, but this is a section through the normal brain. You see the outside has this layer of grey matter. I call it almond icing on the cake. It's about 4 or 5 millimetres thick, it's grey matter. And then we have white matter. The brain cells sit here in their hundreds of thousands, stacked up in special ways. And each brain cell has a long process which goes off and talks to brain cells somewhere else. And those long axons or fibres have a white insulation around them so that they don't lose the impulse as they pass. And so when you put all those white fibres together coming from all the brain cells, you get the white matter. So you see the brain is grey matter and white matter. Now, where we took that section was just through the motor cortex, the area controlling movement on the other side. So if we zoomed in and looked at one of those brain cells, this is what we'd see. Beautiful pyramidal cells. These cells are characteristic of that movement cortex, of the motor area. They have these large branching dendrites. Then they have a fibre, you can't see it, not stained right, but it goes off and talks down finally into the spinal cords and controls, talks to lower motor neurons, talks to muscles on the other side of the body, you see, and controls it. This one brain cell would control just a few little muscle cells, which is one of the muscle cells of thousands which control a movement of your finger. So your movement of your finger, you see, is not controlled by just one of these cells, but hundreds and hundreds of them put together go to make the numbers of cells which control one muscle. And so these brain cells lie on that grey matter, that almond icing on the outside. And if you look at them, you see there's a cell body, and then they have these dendrites going up towards the surface in that layer of almond icing. And they stop just short of the surface. And coming in and terminating on these brain cells, these neurons, are hundreds and hundreds of fibres which cross over, you see. And on one brain cell, one of these typical brain cells controlling movement, you may get upwards of 10,000 to 100,000 terminations on one brain cell to control that movement in that one cell. See what I mean? It's marvellous, isn't it? It's incredible. And you see, looking at this, you don't have to have an interest in science to say that is not only great science, but that is fantastic art, you see. The artistic features here are mind-boggling. The shapes, how they go together, and all the rest of it. And you know, in January of this year, I went up north with my wife, Diana, and we went and stayed in Pai here. And on the way back, we called into Kawakawa. I don't know whether you know Kawakawa, but Kawakawa is a small town, but Hundertwasser, Friedrich Hundertwasser, an Austrian artist, he lived there and died some years ago, and he did this beautiful abstract art. And the toilets in the Kawakawa are world famous. They've got these tiles, all right, which are incredible. So I've never been there before, so we went specifically to see the toilets, okay? And met Italians and Greeks coming out. They had actually visited, they'd heard about this, all right? Walked across the road to the art shop, the Hundertwasser art shop, which showed pictures, abstract pictures, which he did. And I saw this picture on the wall, and I said, my God. I said, what? It's the woman in the shop who knew Hundertwasser. I said, what is it? She said, it's called the blue vase. I said, that's not the blue vase. I said, that's the brain. You see, these are these pyramidal cells, these large dendrites going up. There, it's the cell body. These are the fibres running across, the ones I've just talked about, which make connections. And these are abstract versions of impulses or things inside the cell, of receptors and so on. And I said, I want to buy this because to me, that's the human brain. And she said, my God, I'll never look at that the same. Why I tell the story is that, you see, the brain is artistic. The creativity of the brain is also shown in the creativity of how you can represent it in every way. And, you know, the label on the bottom of this was Imagine Tomorrow's World. And I thought, so now it sits in my office. It's a big print. Everyone that comes in, I said, my God, love your painting. And I tell the story, you see. And that's the story of the wonder of the brain. And that's why I wake up each day and say, I can't get to work fast enough. Because that's what we're trying to solve. And that's your brain, you see. You take it for granted, you didn't have to pay a cent for it. But you've got to look after it and you've got to use it. And you see, and that's the important thing. And how are we going to unravel this? You know, well, one of the tragedies about the human brain is that for many people, 1 in 5, they are affected by a brain disease. And all those cells are not doing the right thing. They begin to die and degenerate. And Alzheimer's disease, you know well. You know, there was a survey done by the Alzheimer's Disease Charitable Trust. Wendy Fleming sitting in the front row. She's chair of the board. They gave money to find out what is the problem that we have in New Zealand with Alzheimer's disease in terms of numbers, in economics and all the rest of it. Well, you know, there are 41,800, I think, Wendy, when you did your survey in 2008, New Zealanders were affected by Alzheimer's disease and dementia. It's huge. Parkinson's disease, another terrible disease. Huntington's disease, epilepsy and so on. I'm going to talk a little bit about these and about some of the things we've found out. Now this is an Alzheimer's disease brain. You might say, well, that's not too bad. But when you compare it, you see, with a normal brain, this is what the normal brain looks like. And you suddenly realise that what's happened in the Alzheimer's brain is that there has been an avalanche of cell death, of cells, especially in this temporal lobe and the frontal lobe. This is the area for memory, you see. This is the area for behaviour and personality. And those cells have died, not 1 or 2 or 3 or 4, tens of thousands. Slowly at first, plaques and tangles kill these cells. Instead of weighing 1,500 grams, a person dying, this particular case, dying after 10, 12-year history of Alzheimer's disease, it weighed 800 grams, all right? Lost a third, virtually, of its brain weight. And you know, if we could cure this disease, it would be incredible. But you know, curing diseases is a huge challenge. If we could do it in stages and just delay the onset of Alzheimer's disease by 5 years, then the prevalence of Alzheimer's disease would drop by 50%, you see. So, doing brain research, we love to find a cure for all these diseases, but if we could just push back and stop the symptoms for years, some years, that would be a great first step. And we've got clues about that already. Parkinson's disease, you see, is a disease which affects different types of cells way down here in the midbrain. Area called the substantia nigra. This is like section through there of the normal brain. We've got beautiful pigmented cells here in the midbrain. Hope mine are like that, which make dopamine. And in Parkinson's disease, you see, they die. And they die very slowly to begin with, until you've lost about 65% of the cells on one side, normally starts on one side, then the symptoms become very clear and obvious. But you've already lost that many cells, you see. You've got to get in early to try and stop it. And then it may spread to the other side. And you get the typical symptoms of Parkinson's disease. We can't slow down that cell death yet. We've got ideas, but we've got different forms of treatment for it, which I'll come back to later on. So that's a huge challenge. Huntington's disease. This is a section of the normal brain I talked about before. And here you have a section through the brain of a person who died with Huntington's disease. You see, this region here has shrunk. We call this the basal ganglia. That's what it looks like in the normal, healthy brain. The person died without a brain disease. This is what it looks like with a person who died with Huntington's disease. Not only has it shrunk down to, you know, under 25% of its volume, but also the cortex has lost brain cells. So it's a huge loss of brain cells. And I'm going to tell you a little bit more about this because it's going to be one of the other things I'm going to refer to about new treatments later. And the thing about Huntington's disease, you see, is it's a relatively rare disease, about 5.6 or upwards of almost of 1 to 10,000 in New Zealand comes on midlife, but the symptoms are highly variable. Some patients show mainly movement disorders, career form movements, some show mood or depression, some show both. So the symptoms of the disease are highly variable, even within the family, which is affected by the disease. And you get different mixtures of it. Some, they have these career form movements coming on very early, and maybe the main symptom right through. Others have mainly these behavioural changes, depression, anxiety, irritability, and only the movement problem comes on later. But what's been known from the very early days when it's first described in 1872 by George Huntington, a family doctor in New York, who took over the practice from his father and grandfather, who were looking after families in this area, and noticed that there was, from one generation to the next, there was this disease which was carried on. And he described it, wrote the first description of it, and said it's a genetic disorder, and it's clearly a dominant genetic disorder. But we didn't know the gene, we didn't find the gene until 1993. And when we found the gene, well, 10 years earlier, 1983, a whole series of studies pointed to that the gene for Huntington's was on the tip of chromosome 4, but they didn't know where it was. It took further studies, incredibly complex molecular genetic studies, another 10 years, and in 1993 they found that the gene looked like this. You see, on chromosome 4, we all have a gene on the tip which is called IT15. It just means interesting transcript or interesting gene number 15. They thought they'd found the gene in the first 14 cases, but they hadn't. You see, so that's why it's called IT15. And then they suddenly found that, in fact, yes, they have located the gene. So this is a normal gene, which is in all of us on the tip of our chromosome 4. And it contains a series of little repeats here. CAG, 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 upwards of 15 or 20 times, sitting right there at the beginning of the gene. In Huntington's disease, for some reason, these repeats just get longer. Instead of being 15, 12, 15, or up towards 20, this area is expanded and it's 36 repeats or higher. And for that one change, the person ends up getting Huntington's disease. It's one single gene change, you see. And it ends up killing cells. It makes that gene will make not a good protein, but a bad protein. And the longer the repeats, the worse the protein. It clogs up the internal cells and they die. And the more repeats above 36, the earlier the onset and the quicker the advance of the disease. We know that now. But we didn't know that until 1993. You couldn't diagnose this condition definitively without a detailed family history and knowing the symptoms, but it could still be confusing without all that data. And so that's another challenge, you see, a huge challenge. And so you see, to solve these diseases, it's really important that you create animal models to mimic the diseases so you can undertake research. You can't actually do experimental studies on the human brain. No one here is going to put their hand up and say, yep, I'm going to go, okay, I'll come into the lab, you can take my head off and away we go. No, it's not going to happen, you see. Ethically, you can't do that, and that's quite right. That's not a problem. So that's why we have animal studies. But what you've got to do, you've got to look at the human brain too, because you've got to look, you see, at this ultimate model, because that's what you're modelling. And sometimes, some groups, I went to a conference where we reported our human cases, and they said, oh, that's all very interesting, what you're finding in the human brain, but our rat models are different to yours. And I said, hang on a bit, I said, you've got the problem, I don't have it. This is the ultimate model. You've got to look at the human brain to find out what's going wrong so you model it correct. And so, you see, when I became besotted by the brain as a young medical student, I took a year out and did research. And of course, I couldn't do it on the human brain. I did it on the rat brain. And found a new pathway and found interesting stuff I never expected to find. Published a few papers and thought, my God, this has got me. You know, my first love, my second love was my wife, actually. I would have got them in the wrong order, of course. She's not really nice. She's more beautiful than the brain. So you see, and then so after I finished the medicine, graduated University of Otago. I tried neurosurgery. I thought that would be really interesting, but I found that you can only do so much. You can only remove tumours and work on the outside of the brain. You can't do much on the inside of the brain back then. And so I then went back into brain research and developed my own little research, did a PhD on the rat brain, went overseas and worked for 3.5 years in Boston and San Francisco, MIT and Harvard, and came back to Auckland and set up my own little lab, just me, the first brain researcher at the new Auckland Medical School, studying the rat brain. And then in 1980, I can remember this vividly, my life changed. And it changed because one of my former professors, who taught me genetics when I was at medical school, Arthur Veal, came in and said, listen, I've got a problem, which I want you to help me with. He said, you know, I look after 400 families in New Zealand with Huntington's disease. And he said, so this is 1980. And he said, you know, the problem that these families have is that there's no test for the disease. We didn't know the gene then, so they couldn't just take a blood test and find out whether they definitively had the disease, because we didn't even know the gene until 1993. He said what they would like is someone to examine the brain of their mum and dad when they died, and often it was the mum and dad during life who actually wanted this request too, and said, listen, would you look at the brain of our mum when after death and tell us whether or not it has this characteristic pathology and shows the clear pathology of Huntington's disease to confirm whether or not the gene's in the family. Because there was the clinical diagnosis wasn't always accurate unless they had all the family history going back several generations. So in my naivety, I said yes. I didn't realise what was about to happen, you see. Because he would arrive along every 2 or 3 months and have literally in a plastic container with formalin, the brain from a family somewhere in New Zealand who wanted to know and unlock the secrets of whether or not they had this disease. And so you see, we would receive this brain. I sat down with a neuropathologist and we do very careful, spend a lot of time, ring up the families, tell them what the result was, write a letter to them, talk to them. They were enormously grateful. And in most cases, virtually 95% of the cases it was yes, and 5% or so occasionally it was no. And saying, no, you don't have, there's no basal ganglia disease, there's no characteristic cell loss in Huntington's, it's something different, it's a form of Parkinson's or whatever. Of course, it's like giving them $1 million, you see. But then those families, you see, what they did, they were so appreciative of that, they just said, look, you keep the brain, and you do research on it, and find out why the brain cells died. And I thought, marvellous. What an incredible, you know. So the community drove our research down this path. And so we gradually, over the years, you see, have collected the brains of people who have died with Huntington's disease. And I call them, the families, the heroes. Really, they started us and they've suffered from this disease in their families, and they've seen their mum and dad go down a line of decline, with each of them a 50% chance of inheriting the gene, and yet they do this, they give this incredible gift to us to do research, to do the diagnosis for them, but to keep it and do ongoing studies. And they became so supportive of these studies, they would write in, and it's usually the donor during life would write in and say, listen, this is what we want to do. We want to give you I want you to have my brain when I die so you can help our kids. Very simple. And so we developed a sort of a donor package, you know, what the forms they'd have to fill in. The donors would sign it and send it all back and so on. And they were so keen for us to get the brain soon after death. Often we got the call from the nursing home. They said, call us first before they call the family when mum's died. Isn't that incredible? And that really humbles you. And so you see, we ended up going back to the families. We found things we didn't expect to find. We needed a family history. We needed to know what the symptoms their mum and dad had, because we found things that weren't written in the textbook. So we collected, you see, the clinical history from the families. We talked to the doctors, the neurologists, and we were given free access to all this information. And so you see, we were having the most invaluable gift. We had been taken down, I'd been taken down a pathway and told to study the human brain and pushed into it by the community, and suddenly we were unravelling riches that we never knew were there. And so our research, you see, is a unique partnership, a partnership between families, doctors and researchers in all sorts of ways, and we tell you many stories. We have family here in the South Island, who whenever they get together, one particular family, in the Timaru area, and they call me once a year when they're all together. They gave their dad's brain to us some years ago, and they said, all the kids are here, they just want to know how research is going. Midday on a Sunday. And I just sit on the phone for an hour. And you think I've given them $1 million. And I tell them what's happening, which I'm going to tell you soon. And that's what it's about, you see. And in 1993, you see, we'd expanded. Not only did we do Huntington's disease, but we had caught the bug, you see. We started doing Parkinson's disease. Families with Parkinson's disease, with Alzheimer's, with epilepsy, motor neuron disease, have come and contributed in this most incredible, unique way. And so in 1995, I went to the Neurological Foundation and said, listen, I think we've got a brain bank here. Because you see, the tissue that we were using, we had all that documentation. We knew all about the brain. We could ring up, and we keep in close contact with all the families. We can ring up every one of those families now, tomorrow, and they want to hear from us, and they come and visit us and see where the brain bank is and so on. And I said, you know, we have established a brain bank here. How about we give it a name? Would you support us? Because we needed money. Because it was being funded, you see, by our research grant. And I said, the Neurological Foundation, you're all about funding research groups. You're the engine house of research, brain research in New Zealand, which we're incredibly grateful for. So they generously said, yeah. So first they funded a technician, and just last year, they extended that to fund the technician plus a research fellow plus all the work expenses. And that's why we call it the Neurological Foundation New Zealand Human Brain Bank. Been funded by them since 1995. Incredible. Thanks, Sue. It's made a huge difference because, you know, what happens now is that, you know, you can do things in small countries you can't do in big countries. We found that when we went overseas and talked to conferences and told them about what we were finding, which is so different to what had been written about the human brain and these diseases, that top research groups said, listen, we would like to study some of this tissue because we can't get access to such high quality brain tissue as what you have in New Zealand. And so we went to the families and asked them, would you be happy for us to send a little bit of mum's brain to the group at Harvard Medical School, to the group at Oxford, Cambridge University in the UK, Gothenburg in Sweden, Zurich in Switzerland, and so on. And asked them if they would be happy to send a little bit of their mum or dad's brain. And in every case, they think it's incredible. They actually are so enthusiastic, and some people have actually said things like, you know, mum always wanted to travel. And she's going to get the travel after death that she'd never had in life. But the fact that she was contributing, or he or dad's brain was contributing to brain research around the world is special. And so you see, they do studies. We only give to top groups who are internationally recognised, who are at the pinnacle, because this is rare tissue. We go for quality, not quantity. We are a boutique brain bank. You see, we're not big like the New York Brain Bank. And so we have to make sure we are the guardians of the tissue. We have to be true to the families and only go with the best, you see. And because of that, we are pushing back the boundaries all over the place. And I say we, our group and all the groups overseas. And that's, the families love to get the feedback. And their enthusiasm and commitment is incredible. One of the families said, you know, we were so excited about this. We went and told our neighbours that Mum's actually going to advance research all around the world. And so that's what the Neurological Foundation Brain Bank has done for brain research here in New Zealand, because of course it's used by researchers throughout New Zealand, here in Canterbury, Otago, Victoria and so on, as well as overseas. It makes a difference. And so what we had unconsciously formed, you see, was a sort of an organisation. And if someone said, what do you want to do in life? And I said, I want to form a big multidisciplinary group with, you know, I couldn't have dreamt of this. And so we had formed an organisation where brain scientists work with patients and families and doctors in partnership with the community to undertake studies on the human brain. If someone said there was no book on how do you make a brain bank, it happened, and it happened because we were forced down that line, a pathway of excitement and imagination by families. And that's why we have such strong links to the community. That's why when Sue or anyone asks me to go and talk to groups in the community, I say yes, because it's payback time, you see. It's reporting back and saying, this is what you have helped us to do. Let us share this with you. And what's really interesting is that when you start doing and looking at the brain in this way, you get unexpected findings. You find out things you never dreamt of. I'm going to just show you some of those things. So we've known, you see, since the 1960s, that the rat, cat and monkey brains, that inside their brains, they still have a layer of stem cells in these adult animals. These are rats running around, cats running around. They still have stem cells left over from when they were an embryo. So when they were embryos, when they were just, the rat was just being, you know, first started to develop, it consisted basically of stem cells which should develop into all parts of the brain and body and so on. And those stem cells are magic cells because they can become anything. Well, it just so happened, you see, is that studies in the early 1960s showed that in the rat brain, so this is the middle of, this is the ventricle here, and there's a right round, so this is the hollow inner part of the brain. There's a lining that goes round that lining inside the brain, this hollow ventricle area. It's called the subventricular zone, this is shown up here. And there's a little layer of armour dicing, if you like, along there, which still has stem cells which are left over from when it was an embryo. In other words, they still have cells that can grow and multiply and make new brain cells. And they make them in a very immature way. They are baby brain cells, if you like. And they multiply and they travel down a motorway. Here you see from Christchurch to Dunedin. And this motorway is a very distinct motorway. And if we go to the next slide, I'll show you a picture of an actual section through the rat brain, which shows us a bit more clear. There it is there. And you see the rat has rats are big on smell. They can smell better than what we can. So they have a huge area called the olfactory, which is a smell area. They're not much good at doing other things, and they can run around, but you don't see them sort of developing computers or developing new ratmobiles or anything like that. So they've got a relatively undifferentiated cortex here, which just controls their muscles, but doesn't make them any extra special in terms of thinking. You have original ideas or writing, you know, E equals MC squared or things like that. And so you see this pathway, as these cells multiply and make new brain cells, they run down this motorway, and they take about a week to get down here to replace cells lost here in the smell area, which is a very active part of the brain. But if you damage a part of the brain up here and mimic a stroke, or damage this area to mimic Huntington's or Parkinson's disease, these little brain cells, immature ones, will leave the pathway and they'll go and repair that area. See, and so lots of studies have been done on this, and It was well established that this is a marvellous facility which animal brains have. They have these stem cells which continue to make new brain cells throughout life of the adult rat, cat and monkey. But the dogma was that this does not happen in the human brain because our forebrain, as you see, has got so complicated and so differentiated. It's a Rolls Royce, you see, compared with the Model T. And we have lost that facility. We don't even have stem cells there. And I was told that, and I used to tell my students that and all the rest of it. Until when we started looking at our human brains, and I cut this short, I won't go into all the details, but just tell you that we actually found evidence that suggested that we also had inside our brain, on that inner area, the ventricles, a layer of stem cells, and they looked like they were making new brain cells. We saw this in Huntington's brains first, but also in others. So when we found this and told the experts, they said, garbage. Especially the top labs in the world who were studying stem cells had looked in the human brain and found no evidence of it. And so we got given a real hard time. And so it became very clear that although we saw in the human brain the equivalent of these stem cells here, making new brain cells, we had to find the motorway. And so we set out, and I put Miles Curtis on this first, and other PhD students on, to look for this motorway in the human brain. And we went about it in a logical way. We took blocks of the brain, we sectioned from front to back, ding, 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 slices through it. We saw little hints of the motorway, but like cutting across the South Island and trying to find the highway going south, you see, from Christchurch. And when you take cross sections, it's not, when you look at 40,000 feet, you can't, it's not easy to find, especially if it's, the highway is a bit small in places. And so instead of cutting, so we found after 2 years of looking with one of our PhD students, and she came back and said, I don't think it's there. I said, it's there. Lied through my teeth. I said, I believe it's there, we just got to look harder. And I said, so Miles was in Sweden, and we got together, and we talked, and his group and our group, and said, we came back and we said, right, let's do something different. Let's, instead of coming from the front to the back, and this sounds so simple, we took huge blocks like this, blocks virtually put into the whole South Island, about 9 centimetres across, and we said, we will section this, instead of from the front to the back, from the midline out. So instead of crossing the South Island, we go along the length of it, you see, and see if we catch the length of the highway, because it will be much easier to see it like that than going in cross section. And so we took literally hundreds of sections, thousands of sections from numbers of brains. To cut a long story short, 3 years work short. When we looked at these sections going from the midline out, we saw bits of staining and sequential sections. And over about several hundred sections, when you put it together, you actually make, and the mosaic is put together, and you can actually see that there is a highway, but it gets very small down here. That's why we were missing it. And that, in fact, you see, when you put it together, this is what it looks like. But there were differences. It got very small down here. And also, it went backwards, not forwards, for a start. But when we put it together, looked at the number of brains, it became very clear this was, in fact, the motorway, but it was different to what we saw in the rat, because our smell area is very small. This part of the motorway, going from Oamaru to Dunedin, you see, is very shriveled up. Not nearly as big as what it is in the rat, but this part is quite large. And it goes backwards and not forwards, and that's why we didn't see it. And it goes backwards because you see the cortex up here in the human has got so large that it's, and this area's got so small that it's pushed the motorway so it's diverted. Instead of going directly from Christchurch to Dunedin, it's actually gone out round through Mount Cook, you see. And it's become distorted. But nevertheless, it is there. And we put the documentation together, we sent it off to one of the top two science journals in the world. One's called Nature, the other's called Science. We sent it off to Nature, thinking, my God, they're going to love this. Well, they sent it out to the top stem cell lab scientists. We believe, we didn't know who they were, but it was pretty clear from the reports we got. We got 3 reports back, and they trashed it completely. And they said, there's no such thing as a neurogenesis or a new brain cell pathway in the human brain. This is absolute, we don't believe this unless you can, the one that actually said, unless you can keep slices of the brain alive and show the cells migrating along the pathway, we won't believe it. It's just not there. So we thought, bugger. And New Zealanders, we do things the Kiwi way, we didn't swallow that. We just put more data together, more information together, repackage it, and sent it off to the other top journal, Science in the USA. They sent it out to referees, and luckily they sent it to referees who did not have a stake in the field, you see. And they said, incredible. This makes biology so clear that here we have the same basic biology in the human brain as we have in the rat brain, but it's different for a variety of reasons. And they said, clearly there's more work to be done. They criticised some parts, wanted more information, which we gave them. And after that exchange, we got it accepted into Science. This is 2007, and we got it on the front cover. And that changed the world. That changed the world. And I would never have believed it myself, you know? And it took, we got attacked. We actually got the major lab in San Francisco, who clearly was one of the original referees and saw this come out, was so mad, wrote to the editor of the journal Science and raised all sorts of questions. The editor of Science sent it to us and said, listen, you're going to have to reply to this. And so that was great because, you see, we could only put so much. Science is such a prestigious journal that you're limited to the number of words and all the rest of it. And we could now reply to it, put all the other information in, the extra radiology data and all the rest of it, and have it out there and prove them wrong. And that's what we did. And, you know, there was still a lot of controversy in some circles, but it's been repeated and shown to be correct. And we could only do this because we had brains from people, from families who had given us human brains to do where we could do these sorts of studies. And suddenly we changed. This is like Galileo, in a way, in a humble way. You know, Galileo was put under house arrest when he said that, in fact, the Earth went round the sun and not the other way round. The heliocentric universe was absolutely opposite thinking, opposite dogma. And then 400 years later, he was actually pardoned by Pope John Paul. And so that was a change in this was a change in the concept now, you see. And suddenly we can make new brain cells. So that opens up huge possibilities, you see. We have stem cells which make new brain cells, and that's revolutionary for our adult. Everybody who has stem cells lying in your brain tonight. And we saw evidence in the brains that if the brain's diseased, it tries to make new brain cells. But of course it's too little too late. We need to make them faster. That's fair enough. But what was interesting is now that we have the same mechanism as what we have in animals, then what they're doing in the rat, you can apply to the human brain. You see, what do we know? Well, we know that if you put rats into a stimulating environment, lots of things to do, paper to cut up, little mazes to run around, and all the rest of it, they make 40% more brain cells. Exercise them, they'll make 40% more brain cells. Over-exercise, stress them, they'll make fewer actually. So apply that to human. The more enhanced your environment, the more stimulating, the more you're using your brain, the more new brain cells you're actually making. Now maybe later on in life you may make a few less, we don't know. We can't do those experiments yet. But the principle's there, you see. So little do you know, not only is this a free lecture tonight, but you're probably making some new brain cells. Right? We should actually charge you for that. But we won't. So you see, creative thinking makes new brain cells. And this seems so obvious, doesn't it? And what's so obvious is what my mum used to say, you lose it, it applies to the brain in every way. The more you stimulate, when you get up in the morning, you feel good. Life's bright, you get solutions. If you feel bad, you sort of get worse. If you get someone says, oh, God, you don't look too good today. And then 2 or 3 other people say, by the end of the day, you're not feeling very good, right? So you see, thinking and being positive actually probably makes new brain cells too. Oh, I think so. So you see, suddenly we've opened up a Pandora's box here, and we're looking and we're finding there are off-ramps which actually go to different areas. And we're studying that, off-ramps that go into this area affected by Parkinson's and Huntington's. And off-ramps which go up to the cortex. We don't know how effective they are or how many cells they're making. Probably not enough to reverse Alzheimer's disease or when you've got a major degenerative disease. But one of the things you can do is try and turn it around, you see. Strokes. We know that if you take people with strokes and rehabilitate them with positive environments, give them lots of things to do, exercise them, give them OT, give them physio, exercise everything that they've got remaining, all the functions that remain, they will in fact repair and improve. In some cases which have been documented, you've heard the stories, you've seen it on TV, where people with some types of strokes have got right back and been able to talk again, been able to walk again. There's a few stories of that. So never, ever, ever accept that brain disease is a final. You must keep stimulating your brain and supporting people with brain disease. Giving people hope. Push back that boundary. Slow down the progression of the disease. May not cure it yet, but suddenly you're pushing back the frontier, you see. Think of that, pushing back Alzheimer's disease 5 years out. And you're decreasing the numbers by 50%, incredible. So, other studies we've done on the human brain. Well, because we work closely with the neurosurgeons in Auckland Hospital, we get brain tissue straight off the operating table with the consent of the patients. And we take it to the lab, the pathologist takes it apart, we take it into the lab, and because it's fresh tissue, we can actually carefully dice it up. This is the work of Mike Draganoff in Auckland, and digest and separate the brain cells into individual units, and you can culture them, whoops, a bit fast there, and you can culture them and then analyse individual brain cells. You're not making a brain, but you can keep them alive sufficiently to actually study them. This is what the brain cells look like from a healthy brain. This is what they look like from epilepsy. We also do it with Alzheimer's tissue pack. And this is what we've developed over the last few years. Mike Draganoff and his team have developed this incredible ability. We've got lines of cells which we freeze down, and then we can unfreeze them and use them to try new drugs. That's our dream for the next 10 years that we want to try and do. Use human brain cells and see if we can reverse the pathological process we see in them. And that's incredible. Another idea is when you've got a disease and you want to try and solve a disease like Huntington's disease, which is caused by a single gene, what's been happening is that they've taken parts of the gene, you know that expanded part of the gene? Those CAG repeats part? There's been lots of studies over the years where they've taken that and put it into rats and mice. Not the whole gene, but it's a very large gene. And they've got rats and mice with this small part of the gene in, which show the symptoms of the disease in a very abnormal way, if you like, but they show advanced symptoms very quickly and often die within 3 months. And they've tried to see if they can then use that to reverse and change what the gene is causing. And in some ways, there are aspects of these models where they've only put part of the gene in to a rat brain, which are a problem. They don't mimic the disease very well. We had Russell Snell come and join us in 1998. He'd worked with the original team in Wales and in Harvard to find that gene in 1993, the Huntington's gene. He came back to Auckland, we got him back from the senior fellowship. He walked in the door of my office and he said, listen, we should look at seeing whether we can actually use and study the sheep brain and put the gene for Huntington's, the human Huntington's gene, if we can put it into the sheep, because the sheep brain is much more similar to the human brain than the rat brain is, of course. You can see that easy. And so it was a dream, an impossible dream. I said, sure, let's do that. Well, cutting a 10-year story short, with funding from the Freemasons, we got to try this. And you see, what we wanted to do was to set up with sheep a process whereby they would replicate not the full extent of the disease, but the early stages of the disease, so that we could try and then use those animals with the human gene in to see if we could develop a therapy to turn the gene off early. We didn't want the sheep to get the disease, we wanted to just show the early symptoms. So what we did, the group in Harvard had isolated and had typical human IT15 genes, which we collaborated with them, and they agreed to give these genes to us, one particular gene which had what we call 73 repeats in. And we had to get a group who was expert in putting this gene into embryos, in the early embryos of sheep. And so we ended up working with a group in Australia, a big government research institute in South Australia called South Australian Research Development Institute, SARDI. We found out that they specialised in doing this sort of thing, not for brain disease, but for other diseases. And they had a big farm, a 6,000 acre farm, just north of Adelaide, right next door to the Barossa Valley, which is very convenient. And what they did, you see, is, we stimulated South Australian merinos with particular types of injection special hormones which made them ovulate, artificially inseminated them, flushed out their fertilised eggs very early, took them into the lab and literally squirted in solution containing the human gene. It was a very crude way of developing, putting the human gene into the sheep, and crossed our fingers and made, and this is making the story very quick and brief, 5 years into one slide here. And we developed a series of lines of sheep which, when they were born, we then tested them to see whether or not they'd actually incorporated the gene. And out of 125 animals, 125 eggs, which we didn't treat it this way, we found that there were 6 animals, and we gave them all different names, each animal, which had incorporated the gene. And some had incorporated in different ways, and it was a problem. And others had incorporated it in a way which the human gene would get switched on and make the beta protein, and it was what we call an ideal model. And we just happened to give all of these 6 animals names at random. Hoosie, Rocket, Kiwi, Taffy, Aussie, and Kiwi. And a sheer coincidence that the Kiwi line worked out to be the best line. So what we've done, we've now raised this flock of sheep, and we have about 500 sheep on the ground, the Kiwi line. They are different ages, and we have found that these sheep develop very slowly the typical features of the pathology of Huntington's and some of the early symptoms. Nothing which is advanced at all. Looking at them, you say, oh, they look normal. But when you look at the brain, we see the protein accumulations we see in the human brain. When you look at their sleep patterns, and we find that they're quite different to the normal sheep. And so we've got a very early model now of Huntington's disease. And for the last 3 years, we've been funded out of North America by a big foundation called CHDI, called Cure Huntington's Disease Initiative. And it's costing them about $1.5 to $2 million a year to fund this. 500 sheep on the ground, keeping them alive and keeping them, looking after them and all the rest of it is very expensive. But this coming September, October, we're going to, now that we've identified that they show the very early signs of the disease, we've got a group at the University of Massachusetts who have developed a technique out of animal studies, and other groups have done a lot of work towards this. You can actually inject into rats a sort of a mirror image of a part of the Huntington's gene into rats which have it. And you can turn the gene down and turn it off almost by putting in what we call an interference gene, an interference RNA technology. It's very complicated, I won't go into the details. So you see, we're going to trial, and anyone here who's got a family with Huntington's disease, you can jump with joy, because we're going to try and see if we can turn this human gene off in these sheep by injecting, when I say we, it's the University of Massachusetts expert genetic group, using interference RNA. They're going to place injections directly into our sheep model and see if they can reverse or stop the disease appearing. Do it early in 18-month-old sheep, and then we'll trace that. It's going to take 5 or 6 years at least to work out whether this is really working. But suddenly, we've got an opportunity, you see. And to do it at the beginning of the disease, you don't wait till the disease is there. If a person was to turn out gene positive, to offer this type of therapy would be incredible. At this stage, you're going to have to inject it straight into the brain. So we've got this huge challenges to overcome, but we've got a new direction. We've got a sheep model in Australia, collaborating with groups in England, the United States, Australia, New Zealand, and we drove this project to do it. And it was the genius of Russell Snell's genetic vision that pushed us down a road which no one else would ever fund because it was too risky. So we've got a little way to go, a long way to go actually. But suddenly, it's hope, you see. And if we can work it out for this disease, Huntington's disease, caused by one gene, then Alzheimer's, Parkinson's, other diseases which are caused by multiple genes, maybe this is going to offer a few opportunities. So you see, we're going forwards, we're not going back. And to be able to get in early with a person who's gene positive and do this would be a dream come true. There's a lot more to do, work to do here, but you know, we've actually got the means and the interest, the international support, and the excitement that we could probably turn this around. But there's still got a long way to go, because it's, you know, it's a challenging business. Every step, but it's a new approach, you see, we never had before. So there's a couple of other examples I just want to show you about new techniques, new ways of treating brain disease, which we don't fully understand, some of them. This, you've heard about this, you know, with Parkinson's disease, we know it's caused by a problem deep inside the brain where these dopamine cells die. And by accident, some years ago, 10 years or so ago, a French group found that when they were going in to do particular treatments in the people with Parkinson's disease, when they stimulated deep inside the brain, in some cases they found the symptoms in a particular area, the symptoms would actually decrease. And if you altered the stimulation in a certain way, you could almost get rid of the symptoms for certain types of patients. This is called deep brain stimulation for Parkinson's. And I've got a clip here of a patient who actually has this deep brain stimulation, a patient with Parkinson's disease. He's going to tell you the story over a short clip of what he's had. You see, he's had the stimulation is provided by an electrode which is placed deep inside the brain in the subthalamic nucleus. The wire comes out and they take it down to a little receptor here, which is a battery-operated stimulator. And you can turn the stimulator on and off, and you can change the rate of stimulation, and you can help certain types of patients with Parkinson's disease. This type of surgery is now being offered in Auckland for New Zealanders. Not very many, it's a start. It's a new type of treatment. We don't know the science of why this works. It's found by accident in a way, but it does. So just watch.
The probe that is placed into the brain, I understand, comes level with the top of the nose and the ear. And it is connected to a battery in my chest, in the skin of my chest here. And this is controlled. I can control the amount of the voltage going in, I can control the length of time that the pulse is, and I can identify the number of times per second that it goes in. And I'll turn myself off now.
So he's turning it off.
So I'm having trouble not only to appreciate that you come to the weeks or person aging, but because my concentration now is up to 90% of my tremor and trying to control it, and I'm thinking the word Parkinson's all the time. I'm not terribly, I'm not concentrating on my conversation with you at all.
Kids are back on again.
Okay. You can see this quite.
So for him, you see, this is a miracle treatment. It's not, we're still developing this. We're still trying to understand. And it's not the perfect treatment for all Parkinson's patients, but for some types of Parkinson's, this type of stimulations and variations on it may help. You've got to tune them. There's a lot of work gone into this. There's a lot more research to be done. But actually, this is where research lags the actual treatment, you see. We've got to work out what it is that you think stimulating the brain in this particular area. There's 2 or 3 areas you can stimulate. And you can get this result in certain types of patients. Miracle treatment. Then there are other interesting developments where you can get what's called medical bionics to actually, I call it magic research, to actually activate. And you know, if you become paraplegic and you have a transection of your spinal cord, so you're paralyzed from your neck down, you can't activate your muscles. But there's some ingenious engineering, electronic, IT type approaches together with neuroscientists looking to see whether they can make machines which can read what you're trying to do in your brain and transfer it to your muscles or to a robot. A long way to go, but let me just show you this example. This person, you see, is paraplegic. And so can't move the muscles. But if you put these electrode arrays and implant them into the motor cortex, the area, which here you see, controls your arm, and you can get it interfacing and actually recording. If you think of doing a movement in your arm and that will activate this area of the cortex, then if you can get that recorded and read out and transcribed back down through ingenious electronic devices to your muscles in your hand, you can actually make your hand move. It's a way out, but there are really positive indications we can do this. I would never have believed it. I don't understand it all. I can't tell you all the details, but this is stuff for the next 5, 10 years, you see. What is interesting is that, you see, when you go to actually when you go to do a movement, you actually dream of it, you generate the idea here, and it will stimulate your cortex here, which controls movement, in a way which stimulates those cells that moves your arm. That's simply what you do. So one thing that's happened, several ingenious research groups have actually implanted electrodes here in your motor cortex, and the person has been trained who's paraplegic, and you can read that out and take it to an external electronic device which will control a robotic arm, and you can think the movement through and make the robotic arm, through engineering ingenious, go and give you a drink. And there is, this is a short clip.
The progress is coming rapidly. They're working on a wireless version of the implant to eliminate the connection in the skull. And Dr. Jeffrey Ling told us that the lab experiments will one day enter the real world.
So, you see, these are dreams for the future. I would never have dreamt that we could achieve this. We're not, you know, when you think and generate an idea, if we could utilize that to help overcome afflictions like paraplegia or whatever, muscle dysfunction, then that would be marvellous. So we're onto a new horizon, you see, where this term multidisciplinary involves not just neuroscientists, but engineers, electronic experts, people who think naturally, who jump in with ideas. And we've got lots of people, different research groups around New Zealand contributing in ways towards this. Most of this is overseas US research and so on. But it's what you're going to hear more of. But it all gets down to the fact of making partnerships, you see, which is what our research has been involved with. Developing team research, forming a creative environment. And what I call the All Blacks of Brain Research, you know. So we formed this Brain Research Club at Auckland that Sue mentioned to you in 2009. It's the Centre of Brain Research, which formalised and brought together the research groups in the university and the doctors, neurologists, neurosurgeons in the hospital and the community organisations. We sort of formed this virtual umbrella, an organisation called the Centre. The Centre's a virtual centre, but we've brought a lot of the scientists together on particular floors and got them working together, not in different departments, but in the same laboratory complex to generate new ideas. And so, you see, we started with 25 groups, and we now got 56 groups. We actually didn't advertise, but it became so successful, this interaction, that everyone doing different types of brain research, from psychology through to genetics, through to molecular biology and pathology, all want to be part of it. And so we've got this club going. We've got more than 300 people, you see. We involve the doctors in the hospital, the neurosurgeons and neurologists, and we're studying to see, can we understand deep brain stimulation, head injury, and do research to actually make them better. and the community partners of all the different organisations who support the Brain Bank, and who are involved in what to hear the latest in brain. They are the third pillar, you see. And now we're going together to bring not only the researchers in Auckland to work better together, but the researchers here at Canterbury, brain researchers here, and also in Otago, to form a big super, it's like Crusaders going to the All Blacks, Anyway, and that's the dream, you see, team research, and it all starts when we suddenly were pushed this way, at least I was pushed this way, by people in the community. And so the mission is to develop treatments for brain disease, and that'll come in all sorts of packages. The challenge is that 1 in 5 New Zealanders is going to get brain disease. Neurological disorders are amongst the top list 5 common disorders. Our goals are simple. They're the same goals, actually, that the Neurological Foundation is giving us money for. All of these groups, virtually all of the groups who are doing brain research, have been in one form or another, helped at one stage or another by the Neurological Foundation, by you people. And those are our goals. They're very simple goals. We're getting there. We're a long way to go. But we're going to get there, and we're going to win. And so collaboration is the key to success, and science is about people. And thank you. It's been great talking to you. Thank you very much.
Breathtakingly remarkable. Agreed? Yes. Absolutely. We're going to have probably around 5 questions, I think, Richard. Richard was going to talk for an hour tonight, and I think he's spoken for about an hour 35. And that is a sign of his huge passion and great interest. If you do need to leave, if you wouldn't mind leaving out the back rather than coming down through where we're going to do some questioning. Now, we've got a very special guest here tonight, Richard. Mr. Pat McGloin has come from Rangiora, and he's been a long-time supporter of the Neurological Foundation. It was also actually the first donor to the Neurological Foundation Chair of Neurosurgery campaign. Mr. McGloin, would you like to ask your question?
Thank you. I corrected my Rangiora as part of my address, but I live in a little country district called Horrell, and I grew up in the Downs, which is nearby, which is a very limited soil type, and I changed to some of the best soils in Canterbury, but it didn't alter the fact that when I was young I had what was known as the goiter trouble. In those days, this is in the late 20s, early 30s, they used to just give you a sea meal on porridge. And ever since I've only eaten porridge if I was out somewhere and my host presented it. And as good faith, I've never mentioned it, but I've consumed the porridge. But the extraordinary thing at that time was that they used to paint your throat with iodine. And my mother was a very proud person, and she made certain that she only used white iodine. She didn't want to have anybody have a blotch on their scutcheon in those days by being prepared to have suffering like everybody else did, I suppose. But anyway, the interesting thing to me is the environment when I changed over. I've always bred horses, that the foals on the good soils that I went to still had developed goiter. And the only way you could do that was by presenting the mare with a better class of feed in the form of salt in the diet, iodized salt. So those were sort of simple things, and it occurred to me that somewhere or another in all of this, there must be an influence of the environment as well as on the end product, which is tonight I found quite fascinating. And I'd love to go to South Australia again and see those sheep. And
Well, you know, you've made a very important point, which as scientists, I was brought up and thought that you had to be able to explain everything in order to believe it. And, you know, genes, I was taught that, you know, at this gene, you'd have genes for particular things. Well, we know it's more complicated than that. And that genes, you know, the Huntington's gene, for example, interacts with in each of our cells, we've got 45,000 genes, right? And the Huntington's gene actually affects the activity of about 20,000 other genes. And so it's actually not a single gene disease, it's a multiple gene. And also your environment, the expression of all these genes, so genes don't work by themselves, they interact with each other. And your environment can actually have significant effects on your gene activity, what's called epigenetics. And so in Huntington's disease, they've worked out that about 60% of the progress of the disease is due to the gene, but the other 40% depends on environmental factors which are altering the expression of those genes in the bad gene. And so suddenly, you know, nature and nurture is very important. What is written in your genes is not the ultimate story, it is part of the story, and that you can alter and change your gene expression by your environment in ways we just don't know, by the way you interact with people, by having a positive or negative outlook or whatever. I say that in the nicest possible way. You know, you can turn yourself down and you can actually influence your genes. So science is complicated, and so are diseases complicated. If you take a person with any illness and give them care and attention and support and give them hope, you will slow down whatever disease they have in a significant way. You won't cure it, but you will change them. And that's what we've got to remember, you see, environment, you know, Alzheimer's patients. We can't cure and stop the first cell dying, but if you put them and support the carers that support them, and give them environments, it will actually slow the disease down. It's not rocket science, but it actually does work. And that's one of the things we're going to look at in our new research program, which we're joining up. We're going to try and see, not only look at the science of how we can do that, but turn the gene off, but also look and see how we can change support and care for patients with brain disease to alter the progression of the disease. We'd love to cure all the diseases, but if we could just slow them down for a start, that would be incredible. And often people with the most impoverished circumstances, with the most severe diseases, can actually slow their disease down. I could go on, but I won't.
Thank you, Richard. Have we got a question on this side? Question up through the middle? Richard's answered them all. Okay, any questions over the other side? No? Not one. Oh yes, we've got one here. We'll take these two, Richard.
Okay.
I think everyone here tonight's been just blown away by your presentation. But I wonder what you can advise us to do, each of us in this audience, that would help the research that you're doing, something that we can do apart from giving money to the Neurological Foundation. Is there anything else we can do that can help you?
Well, you know, one of the ways is, and we're doing this in our centre in Auckland, but I know it's also done with the researchers here at the New Zealand Brain Institute here and other researchers in Canterbury, you know, is we're interacting with people who've got brain disease and doing clinical trials and actually doing studies of them is really important. And that's one of our major thrusts that we're going to do, is not only studying in the lab, but also study people during the course of the disease. So when, through the Neurological Foundation or through these new organisations, We're getting registers of people who have diseases who would like to become involved with possible trials, and I don't call these experimental trials, but just monitoring the progress of the disease, then seeing if we can turn it round by certain things, by comparing a control group with an experimental group or whatever, is going to be really important. Supporting Neurological Foundation, of course, is very clear, and they support our brain researchers. You know, brain research and science in New Zealand, we get the lowest per capita of GDP of any OECD country in the world. Isn't that incredible? And yet we are a country of innovators, and in a way, and I want to say this in a positive way, it's a shortage of funds sometimes that makes us get ingenious, but we actually do need funds, you know. So, but we collaborate and are driven to collaborate by the fact that we've got to make the dollar go as far as we can, but we actually need a lot more support. So look, and where there's opportunities to support research drives in areas you have got interest in, just put your hand up. Neurological Foundation website will give you those. Go to the Centre for Brain Research website in Auckland, and others for the New Zealand Brain Institute here, and so on. And I'm sure the Neurological Foundation, in fact, Sue, can I put a challenge to you in the next newsletter, where are you, to actually put a list out of how people can support brain research in different ways, a practical list. And they've probably already done this, but reminding you that in fact there are ways, because it's really, we need the involvement of people to help drive it forward. And also, but giving support to groups, Alzheimer's, Huntington's, Parkinson's. You know, Alzheimer's disease, the Alzheimer's Charitable Trust is raising money to help and try and see how they can help looking after people and creating better environments for people with Alzheimer's disease. Putting them all in one place is not necessarily the best thing. Providing different types of support. And Wendy, I could ask you to talk, and you'd probably just, you know, say there's so many things to do. But so support these organisations, because New Zealanders are great at support. The actual, you know, patient support for clinical trials amongst New Zealanders is some of the best in the world. And despite the fact we are underfunded, despite the fact we've got to rely on Neurological Foundation with very little support from the government, the government money going into research overall, and brain research in particular, has not changed for the last 10 years. It's actually gone down relatively. And so for people to go into brain research and have a career, you actually got to put your life on the line. For people who've got families and so on, the most you get funding for 3 years at a time. So those are some of the cold, hard facts. But in the next newsletter, Sue will give you practical examples of how you become involved, all right? But your involvement tonight is just great. Thanks.
Thank you, Richard. I accept that challenge. We've got our last question of the evening.
Hi, Richard. Thanks for coming to talk to us tonight. I'm 22 and I've got friends at the University of Otago at the medical school, and some in Christchurch as well, who are really interested in this sort of work that you're doing. Obviously sort of aspiring to help out, but having done nothing themselves. You spoke about it was really exciting to hear about the Centre for Brain Research and this sort of collaborative approach that you're kind of pushing. And at one point you talked about getting Canterbury involved.
Yep.
I'm just interested to hear what students of a similar age at the University of Canterbury might be able to do to contribute to your work. I'm doing a medical degree at Otago.
Well, get involved first, take papers in neuroscience at the university, talk to the lecturer, see what research projects are going on. We're collaborating with groups here. University of Otago, they have got a brain health research centre. Director's Cliff Abraham. They've got a group similar to ours. They've got different types of strengths. We sort of complement one another. So that's why we want to get together with the people at the University of Canterbury. And we're trying to do it in a way that's going to get this extra funding from government. But just do papers involved with the brain during the undergraduate years, talk to the people, and you'll find each of the lecturers has got a research program of some form. engineering, you can see getting engineers involved is critical. Electronics people, this stuff's way above my head. God, to think, thought, putting an electrode in your brain and thinking an idea out and being able to drive a robot for the fairies. Well, the principles are there, so we need people going into electronics and having a neuroscience interest and crossing the boundaries. So there's, in your engineering school here, there will be opportunities. There are people, and there's some top scientists here who are modelling brain cells. So the opportunities are here in Canterbury. It's a great university. You people have actually ridden out the biggest challenge of your life, an earthquake. God help me. And these challenges in life bring out the drive to go forward. And so Canterbury University is having a lot of challenges at the moment because for all sorts of problems. But you've got great scientists here. And so doing undergraduate papers and going into either engineering or neuroscience of some form or another, there are opportunities there, huge opportunities. And enthusiasm, passion and commitment, we can do anything.
Thank you.
I interviewed a scientist at the University of London last week, a couple of weeks ago actually. Richard knows who I'm talking about, Emma Stotter, Emma Daniel. She's a motor neurone disease research biochemist actually. And when I asked her who had influenced her research career, and she is an absolute dynamo of a scientist, she said Professor Richard Faull, and she also added that he had probably deviated a lot of would-be cardiologists and paediatricians from their prospective careers. They started out first, second year med school, met Richard Faull in the neuroscience papers, and away they went. And it's probably appropriate to mention also that Dr. Maurice Curtis, who was mentioned as the PhD in Richard's Science Journal publication, was going to be a radiologist and met Richard in his first year. It's been an absolute honour to present Richard to Christchurch and Cantabrians this evening. Richard was going to talk on February the 25th, 2011, and unfortunately was not able to speak. And it's been a long time coming, but I hope it's been worth the wait. Richard, you're an absolute an inspiration to all of us who work with you, to all of us who are affected by neurological disease, either through family, personally, friends, and it is incredibly remarkable for us all to have the knowledge that you're at the helm of neurological research in New Zealand, leading the way, and you're never going to retire, okay?
Thank you.
Would like a certificate of attendance. We do actually have some out the front. We've got around 60, so if there are more registered nurses, enrolled nurses, carers, support workers than the 60 that we have, we're able to take your name and address and mail one to you. This is what they look like. If you do need it to be signed, then just ask one of the Neurological Foundation team. Thank you so much to our volunteers this evening, and thank you very much, all of you, for coming out on a Wednesday night in the rain. We greatly appreciate your support. Thank you very much. Travel safe.